Welcome everybody. This is our science and technology series and this is the part 2 of the biotech lecture. So as we have seen in the last lecture, we have covered the basics and the related terms. This lecture we will be starting directly from the branches. Branches of what? Branches of biotechnology. So it has various branches which are given colors like red biotech, green biotech, blue biotech, white and yellow. Now these are given such colors with respect to the field they are in. So it becomes easier to remember which color relates to which field. So let me just quickly explain them to you. In this you just have to remember the classification and the field they are in and example of that field. All right, so in the way I have brought this diagram for you, in that similar way you can read them. So red biotech is basically, it is the biotech which is used in the medical purposes or medical field, such as gene therapy, right, cloning, stem cell, DNA profiling, vaccination and so on. So these are the applications. Already you understand biotech already, so you don't need to understand how basically we are going to apply it. So basically in medical biotech we are going to change the biology of any organism and create products out of it, products like gene therapy, stem cell, etc. Now when biotech is applied in agriculture field, it becomes green biotech. Here you can use biotech to create genetically modified crops. You can create bioremediation, like you can create a bacteria which can protect against weeds. You can create biofertilizers, you can create biopesticides and hybridization. Then what is the blue biotech? It is basically use of biotechnology in marine and aquatic applications, like protection of certain species, oceanic exploration. Basically blue biotech is, as you all know, the sea creatures, they have adopted to live in very difficult weather conditions and they live under high pressure of water and so on. So basically biotechnology of the blue kind, it is concerned with the exploration, right, of the diverse marine environment in order to develop new products. All right, so basically oceanic exploration is there. You must have heard about omega 3 capsules, right? So such medications are also derived from this blue biotech only. Then white biotech. All right, this will be biotech. Here you use the microorganisms, various microorganisms to produce some things like bacteria, you use fungus, you use enzymes to produce some products like curd on industrial level, wine on industrial level, cheese on industrial level, beer, idli, vinegar, etc. So whenever you use these biotechnologies or you use biological products to create something which is produced at industrial level, so that is the white biotech. Yellow biotech is use of insects for our benefit, like you can alter the gene of some mosquito to stop propagation of some disease, like vector borne diseases are there. Then pollination, you can create such, in the future maybe you can create or modify the adaptability of honeybees and so on. All right, now we come to the application. So that is all you need to know in the types. Application, first of all, environmental application. One of the most important is treatment of waste. For example, Pseudomonas putida. This is basically an oil eating bacteria. It really eats the oil. So it could feed on oil, and which could obviously help in oil spills, right? Also it can help, you can basically use this idea and use or create other bacteria in order to make some bacteria which can actually eat other type of waste. This is just an example, and this was all, and one of the, why I'm taking this example, because this was created by an Indian born person, Anand Mohan Chakrabarti. So quite a good example, and if you write the name, it is well and good. If we apply biotech in industry, it can be used as, I've already discussed this, for example, to produce alcohol, and then sweetening agents for soft drinks, to produce cheese, to produce wine, right? In health, it can be used in diagnostic kits. Like right now, biotech has been used, the technology created by biotech has been used in COVID-19 RT-PCR tests. This part PCR, I've discussed about this. Then in creation of hormones also, like insulin, growth hormone, all right. So what happens is we use a bacteria, we change the we insert something in it, and then bacteria produces insulin, all right. Then in animal husbandry, like you can create and inseminate buffaloes to create healthy offspring. Embryo transfer is there, all right. All these things are used to breed better animals who have good qualities like high strength or better output, or basically milk or better meat and so on. So basically, if you haven't heard about it, then dogs are also bred artificially to create new breed of dogs. So like the Vodafone dog, right? That dog is a artificially bred dog, like breed. So all these things are done because of artificial insemination. Then if we come to agriculture, then here I have discussed some things with you like GMO and transgenic plants. I have already discussed about this. Then biofertilizers, basically here bacteria will be used as a fertilizer, as a part of fertilizer. That is the biofertilizer part. Then pest resistant plants, we have talked about this, that we can create a plant which can become resistant to pest, which is BT cotton. The bacteria's part, like the BT part is the BT part comes from where? From bacteria only, na? So BT cotton, in which BT is the bacterial part which makes the cotton pest resistant. Then one of the most important application is in medicine, of course in agriculture also, but the one of the most important things that we face in day-to-day life is medicine. So here there are many things like gene therapy or gene editing, we have discussed about this. Then rDNA technology, I have also discussed about this. Polymerase chain reaction, PCR, RT-PCR test, currently being used in COVID-19. In animal husbandry, one thing I would like to tell you is that Indian scientists have developed basically a procedure to produce several identical buffaloes by using nuclear transfer technique. We will discuss about this later on also, but briefly let me tell you, nuclear transfer technique basically means you transfer nucleus from a donor cell to a recipient cell, and then this recipient cell will propagate. So you just transfer the nucleus from cell A to cell B, in which you first remove the nucleus from cell B. All right. In medicines, biotechnology can be used a lot, right? It is being used a lot, and it will serve the purpose of early diagnosis. Lot of early diagnostic kits are being developed only because of biotechnology. That means there are many tests that can be now done to detect diseases before they develop into a serious condition. Now, since we are studying application, we will study few of the specific examples which are very critical for examination purposes. Many of these things have already been asked. So in UPSC, how you prepare is like this only. You see previous year questions and then you prepare the related topics. So here in this section, I have brought for you many things which are related to the things that have been already asked. So we need to, obviously we cannot go into depth of every application, but we will be studying what are most common and basically try to cover as much as possible. Because in general studies we cannot go in a lot of depth, that is not required. So the first topic that we are going to see is gene silencing. Just see the name, gene silencing. So obviously you are going to shut some gene up, right? So here basically you shut down a gene. We have studied about gene, it's a part of DNA that does something particular. So this gene will stop doing that particular thing. Alright. So in DNA, you shut down a part of DNA by using gene silencing technique. So basically you switch off. So what will happen is, let's say gene A will, or it used to perform some function. So by using gene silencing, you will stop it from performing that function. Like let's say that gene is leading to some medicinal problem in the body. So you will deactivate that gene, so to speak, and then it will not lead to propagation. That gene will not lead to any propagation. Alright. So under normal conditions, it will express itself, but if you use gene silencing, it will switch off. Now, how do doctors do it? They use techniques like RNA interference, they use CRISPR-Cas9 and antisense RNA technique, by which you can suppress, you can really stop a gene from producing any proteins, or basically giving any instructions to produce any protein, right? So very easy concept, and practically this has been used. Researchers from ICRISAT is an organization in Hyderabad. It stands for the International, I'm writing in short form, International Crops Research Institute for the Semi-Arid Tropics. Alright, International Crops Research Institute for the Semi-Arid Tropics. Now it is located in Telangana, Hyderabad, sorry. Yeah, basically the same thing. So ICRISAT researchers make peanuts free of aflatoxin. Now aflatoxin is a toxin, basically, obviously it's in the name. It is produced by a fungi. Now what did they do? Let's see that. So they used two techniques in which they inserted two alfalfa genes. Alfalfa is a plant, right, which enhances immunity of a particular plant against the fungi, which causes the toxin, which produces alpha toxin. So basically they inserted two alfalfa genes to give immunity. Alright, basically alpha toxin is a toxin, right? So to prevent the plant from getting toxic, they gave it immunity. And they gave it immunity how? By inserting two alfalfa genes. Now this is genetic modification, and this is how you can also understand. So step one was genetic modification of the groundnut, right? We are studying about peanuts. So the second step that they did was they prevented the production of alpha toxin if an infection happens. Now let's say the alfalfa genes did not work. They did not work. Let's say in the worst case scenario. So what will happen? So as a backup, they created, basically they silenced a gene, all right, which produces alpha toxin if there is a infection. So basically first they granted immunity, then they silenced the gene because of which the alpha toxin is produced. Okay. The first technique they used was genetic modification. The second technique they used was gene silencing technique. Alright, so this is done in India, so this is a good example to take. And one of the best thing is it has there are two examples of technology in this example. I hope you have understood. You don't need to remember the specific things, but yes, I have written the minimum things and which these should be remembered. You don't need to go and read the article anymore. These are the minimum things that you need to remember. All right. Now we have just heard about this term RNA interference. Now interference means basically stopping something from happening. So scientists, what they do is they send something which stops the instruction from going on from the messenger RNA. Very basically, I just want you to understand, that's all. So basically, instruction would be sent from an RNA, and by using RNA interference, they stop that instruction from going on. So this is a very exciting technology by which you can stop basically silence a gene. So you can actually cure somebody by stopping the production of protein in their body or production of cells in their body. So this basically is used and will be used for disease prevention. So RNA interference basically happens when you use the own mechanism of your gene to stop it, or production of the you can say message. So I know actually this is a very it's a bit complicated thing, so not very easy to explain. So point is, the messenger RNA, you have to stop the messenger to deliver the message in order to develop a protein. So what happens is every gene leads to development or production of protein. So you have to silence that gene if you want to stop the production of that protein, and humans are basically made of proteins. So that is the technology here. All right, that's all you need to understand. Now what I have written in the slide is quite technical, so you can read it, and whatever I have explained now you will be able to easily understand, but for your benefit, let's just read it once. The term RNA interference was coined to describe a cellular mechanism that uses gene's own DNA sequence of gene to turn it off, a process that researchers called silencing. Now it can be used in animals, plants and fungi, and it is triggered by double-stranded RNA. All right. So basically, the doctors or any researchers will send an double-stranded RNA to silence a gene, and they will be sending it in a body. So what is the benefit? Basically, you can create genetically modified crops. Then you can cure lifestyle diseases. You can reduce food wastage. You can treat cancer and AIDS. Basically, you can identify the gene that is causing and obviously silence it. So in genetically modified crops, you can create genetically modified crops like BT brinjal, BT cotton and even mustard, but they are created using DNA technology, not RNA technology. So in the basically their DNA is changed, their gene is changed, but here you can use the original DNA and just silence the gene. So basically you can just stop it from producing something, that's all. All right, it's like telling a human not to swim in the ocean if they don't know swimming. All right, rather than giving it gills. So that's the thing. So you can create genetically modified crops, and hopefully if you don't change the DNA, you will not meet so much resistance from the non-G or basically anti-GMO, right? There are anti-GMO protests. So if you create something with the help of RNA interference, you might not face the same criticism or resistance. Secondly, how you can treat with or how you can treat lifestyle diseases. Lifestyle diseases are like obesity, hypertension, cancer, heart diseases. All right. So you can basically there is a good alcohol good cholesterol and a bad cholesterol. So you can use genes that can reduce cholesterol, and you can silence them to reduce cholesterol basically by using RNA technology. then you can use to or basically you can use the silencing technology to create or increase the shelf life of a food. There is a breed of tomato called flavor saver that was created by antisense technology, and it has increased the tomato's shelf life. So basically you won't need so much cold storages, and this is very good for India. So if we can create such things in India where you don't need to store them in cold storage for so long, because already infrastructure is in a quite limited sense in India, it will benefit us. All right, so I just mentioned antisense technology. So what is that? Just a small difference from RNA technology. Like here in RNA, I said it is triggered by double stranded RNA. So double stranded means, just a second, like this. But in antisense technology, it is just the single stranded RNA. Just that is the difference. So whenever you talk about interference, you can talk in the same sentence about RNA interference and antisense interference. They are same technologies, right? So antisense technology was used to produce a variety of tomato which has higher shelf life, known as flavor saver tomato. Now here you can see the depiction. Traditional tomato would need sealed packaging with a gas to prevent spoilage. But in the modified version of the tomato, you don't need any such packaging, you can directly send them. So this is a good thing. Here you haven't changed the DNA of the tomato, you have just changed the or silenced the gene that produces basically old age. So even humans, it might be later used to prevent old age. Again, human testing is obviously banned, and it is a very ethically concerning issue. So right now there is no testing on humans. All right, now we come to a very critical part called as somatic cell nuclear transfer. Somatic cell nuclear transfer. So let us break this down. Now if we try to understand, I have already told you about nuclear transfer. So in nuclear transfer, you take a nuclear from one cell to another cell, but first you remove the nucleus from the cell B. So there is donor cell and recipient cell, and now B will propagate but with the nucleus of A. All right, what is somatic cell? This means any cell of the body, any cell of the body, but not the germ cell. Let me explain. Germ cell is basically the sexual cell that produces sperms in male and eggs in female. All right. So basically germ cell is the cell cell which produces sperm and eggs. So anything that does not produces sperm and eggs is a somatic cell. So somatic cell is any cell that is not a germ cell. For example, brain cell, kidney cell, heart cell, your skin cells also. All right. Okay. Now in somatic cell nuclear transfer, this A cell will be a somatic cell. All right. It will not be a germ cell. So somatic cell and transfer of that nucleus. That's it. Now this B1, this B will be the new cell, and this will propagate, and then it will be a pluripotent cell, and then it can be used for anything. It can be used to for therapy also. It can be used for as we'll see cloning also. So what is the process? Basically very simple process. A somatic cell, basically I'm talking about creating a child. All right, or how this cloning happens, or you know how a birth to a child is given, basically IVF technique. A somatic cell is isolated and extracted from an adult female. The nucleus of all its DNA from an egg cell is removed, and then the nucleus from the somatic cell is transferred to the egg cell. After being inserted into the egg, now when you have completed the transplant, that is the nucleus from the somatic cell, that is the cell A, is inserted into the cell B. You give a shock, electrical shock to cell B, and then it produces more cells like it. Alright. So this cell B will behave like a freshly fertilized egg. Now it will develop into an embryo, and then it will be planted into the surrogate mother. Right. So this is the process of surrogacy. Now here see the diagram. Somatic cell. Alright, this is the egg cell. Now somatic cell, you will take out the nucleus, and here you will take out the egg cell and you will remove the nucleus. Then you will combine them both, and then you can propagate the same cell. Now when you do reproductive cloning, alright, so this thing, okay, you will insert into surrogate mother. When you want to do therapeutic cloning, you will create more cells like it and create a tissue culture out of it, and then use it according to your own needs. Now we have seen the reproductive cloning. Right now cloning is not that cloning, human cloning, it's basically cloning of the cell. So we will see the therapeutic cloning right now via diagram. Again, we don't need to go into depth, we just need to basically know what it means. Now let me explain human cloning. Alright. So basically human cloning means cloning the cells. That's all. So it's not that cloning again, but it is close to that. Basically you clone the cells. So you take it from human. Alright. Then you same process. You combine everything, and this is the embryonic development in the lab. That means first of all, see this this one. You take out the somatic cell, then you take out the nucleus, then you plant that nucleus in an egg, alright, and you have to remove the nucleus from the egg, the original nucleus. Then this become egg with somatic nucleus, then you activate it by shock, and then you develop it into lab. Now this you can use to create a child, or you can create a therapy. Basically you can create cells that can be used to create therapy for humans. So now that we have understood the same thing from two different diagrams, let us understand the terms discussed. Surrogacy, basically what it means. So here you use a woman's uterus to basically carry out a pregnancy, right? But that pregnancy is for some other couple. That is not the woman's own child. Alright, so the woman who carries the child is the surrogate, and the child will be of the parents. Right, in vitro fertilization is used to place the fertilized egg of the parents into the surrogate's uterus. So this is the first part. Second is therapeutic cloning. Here what do you do is you clone embryos. Sorry, not clone embryos, you create a cloned embryo. Alright, which will produce stem cells, and these stem cells will be same as the donor person. Now let's say I have a disease, right, and science has achieved this stage where I can or you can replace the organ or create a heart, basically same as mine, or basically create another human as me, right? So cloned embryo, in this you create in therapeutic cloning you create a cloned embryo only for producing embryonic stem cells. Now these stem cells will be used in the future, I'm talking about, will be used to cure some disease in me, or even create somebody like me. Alright, so the cloning part has never been tested till now, in which a same person will be created like me with same physical features. Alright, with same body structure, with same muscle tone, and so on. So what is being done right now? Right now these cells are being created, but only for understanding some diseases. Like you create a cell, see stem cells can be can change into anything, right? So we are trying to understand how it develops into a disease. That's all. So there are two kinds of human cloning, therapeutic and reproductive. The reproductive one is the one where you create a proper clone of a person which has not been tested. Therapeutic ones are being used to create some solutions for the diseases. Remember that. Therapeutic cloning is cloning of the cells for diseases. Okay. Now we come to an interesting topic, which is a three-parent baby. This is also called as mitochondrial transfer technique. Mitochondrial transfer technique. So in exam this can be asked as a term. So what is the need of it? Problem happens whenever a child might inherit some disease from the mother, especially some disease which goes or basically gets severe with time, especially with respect to your body functions and your mental capacity. Like there is a disease called Parkinson's disease where you lose the control of your body. So such disease happen because of the faulty mitochondria in the mother. All right, from the faulty mitochondria in the mother. So what do we have to do? We will basically, all right, we will basically remove the faulty mitochondria. We will take another healthy mitochondria and put it in the egg. All right. So the faulty mitochondria will not be passed to the child. So listen carefully. So why it is called three parent baby? So let's say the original mother has the faulty mitochondria and the donor does not. So you remove the mitochondria here and you add mitochondria here, then of course the male sperm is there to fertilize the egg and then the child is born. This is how it is. Getting my point? This is how it is. So you remove the faulty mitochondria so that it does not pass to the child and so that the child will not have the degenerative neurological disorders. So how it is done? Process I have already talked about. So what in MRT, mitochondrial replacement therapy, the doctors fertilize the egg from the affected woman, that is the faulty mitochondria of the mother and her partner, right? So husband, wife, or whoever it is, right? So a sperm is fertilized using normal IVF technique. Now what happens in IVF? Another related term. In IVF, the fertilization of the egg happens in a lab. Usually it happens inside the woman's uterus, but here it will happen outside the lab and then it is implanted into the woman. All right, the fertilized egg. Sometimes what happens is the fertilization does not happen in the woman's body due to some or the other medical reason. So this fertilization is done outside the body and then the fertilized egg is inserted into the body. This is IVF. Now till here the steps are same, but after the fertilization, the fertilized egg or the fertilized cells will not be inserted. The faulty chromosome will be removed from this fertilized cell and the donor cells will be introduced into the fertilized, and now it will be basically implanted. So one different step is added. Basically in IVF what happens? Mother, or let's say mother, father, fertilization and then insert into the body. This is normal IVF. All right, insert into body. Now in MRT what happens? Fertilization, but here add donor plus remove mother's genes, all right, mother's chromosome basically, and then insert. So this is the procedure. Let's see in a diagrammatic way. So in this, repair of the cell is done before fertilization. So here is the mother's egg where there is unhealthy mitochondria. If you see the first image, all right. So mother's nuclear material removed and kept. Now donor's egg where healthy mitochondria is there. Now it is that what happens here is donor nuclear material removed and destroyed. So the nuclear is, or the nucleus is taken from the yellow one. Yellow one is the mother one, and then healthy mitochondria, yellow again, small yellow you can see, or orange, right? So bigger orange is the nucleus from the mother. The smaller, the bigger orange circle is the nucleus from the mother, original mother. Then the smaller circle is the healthy mitochondria from the donor that is taken, all right, and then with the help of sperm it is fertilized. So mother's nucleus placed inside donor egg and fertilized, and then the child is born. Second is pro-nuclear transfer. So repair is done after fertilization. Parents fertilized egg, it has the unhealthy mitochondria. So parents nuclear material removed and kept. Healthy mitochondria, donor nuclear material is removed and destroyed, and then parents nuclear material placed inside the donor egg. All right. So basically I described this one, pro-nuclear transfer. So another method is there which is called material spindle transfer, which is the first method. All right, again you don't need to remember these, but you can remember the name. Pro-nuclear transfer, already asked by UPSC. Material spindle transfer. Right, so see, this was the baby which was basically born because of the three parent concept. So the doctor was John Zhang. All right, so yeah, this is the photo of the three parent baby. So what is the issue that is solved by this procedure? So mitochondria is something which actually gives power to the cells. They provide, basically they are the nuclear power plants of our body, or thermal power plants of our body, right? So if the cells do not produce proper energy, they will not be able to function properly, as simple as that. So mitochondrial diseases start as soon as you start growing up. So they strike you in childhood, and slowly they get worsen, or they get worse by age. For example, like I said, Parkinson's disease. Also these affect everything, right? And mostly this is fatal, like people die before reaching 30 or 40 even. So the part of body that need most energy are like what? Your brain. Your brain is so small, but it eats the largest amount of energy, if you know, right? So brain, muscles, your daily functions will be impaired. Like in Parkinson's, you don't have control over your body and how it acts, so that is also affected. All right, then other problems are also there. Your eye, the power of your eyesight will fall, maybe your digestion power will fall, and so on. All right, liver might not produce enough insulin. Oh sorry, pancreas produce insulin, just pardon me for that. All right, so this is the thing, this is why mitochondria is very important. It's simply like, imagine if your state's power plant stops functioning, obviously people will not be able to do their jobs. So if the body's power plant stops functioning, then imagine the body can't work. All right, and this cannot be repaired. The worst thing is it cannot be repaired, it can't be even managed. If it starts, you're gone. It will come for you. This like death comes for us all. Similarly, this disease sadly comes for you if you have this genetic condition. All right, but what is the issue that people have to such procedure? Why? So one of the most important ethical concerns in any medical procedure is consent. For example, this was also the case in euthanasia. Consent is the issue. So consent of what? Of the unborn child. If you change something in the genetic structure of the child, and maybe it solves one problem, but maybe it creates another problem. What about that? Then the child might have to live with the second problem for their whole life. But did you take the permission of the child? Many people say this, that many people, the people who have concerns, they say that have you taken the permission, have you taken the consent from the future person? Of course you can't. So the point is, without their consent, you are making changes in their DNA structure. Also, second objection is, with any technology which is growing, mitochondria are also something which are not completely understood, right? So if you change them, and if you do it, let's say, improperly, they might lead to something which you can't even imagine, right? Let's say the person is driving and suddenly something happens and their eyesight is gone because of the malfunctioning of the mitochondria. Then what? Who's responsible? Things like this. So we need to better understand mitochondria and create safer ways to implant it or change it. Here I would like to tell you one thing. They tested the baby's DNA and found that only 1% has come from the mother, and the issue is only 1%. Like 99% of the issue was resolved. This, I mean, the mitochondria, only 1% issue is there, and they say that to create issues in the body, the error should be at least 18%. So error was only 1%. All right. So the let's say the operation worked in case of this baby. Right. Now, what are the implications? See, implication means what are the observations that we can study. So the mitochondrial DNA is not like the core DNA in cells. It is different. Hence, it will have no impact on the personality. See, it's like it doesn't matter from where you are getting the electricity, right? In your home, it doesn't matter. It doesn't matter if you are getting it from a nuclear power plant or a thermal power plant. You just want electricity. You just want your computer to run. You just want your TV to run. You just want your mobile to charge. So simply we can relate it with this. We don't care from which mitochondria it is coming. It's not changing the DNA. It's not, see, type of electricity, if it is coming from nuclear, it's not going to charge your mobile faster, right? Similarly, the mitochondria, if it is from the donor, it is not going to change the way you are. That's your personality, right? The personality, the way you think, and etc. So it doesn't matter. Then the first thing is this. Then second thing is that here with this procedure you can stop basically passing of genetic diseases. All right. Third thing is, people are worried that if such techniques become very prevalent or very common, then some people might use this or such technologies to create a designer baby, or basically to create a genetically modified baby. That means parents will say to the doctors, we want a baby of such height or such eyes color and so on, right? So this thing is important to know, that concerns of genetic or genetically modified babies come from this. Like we are, why did I laugh? Because genetically modified organisms and genetically modified babies, some similar terms. So this is the thing. This genetically modified babies issue comes from here. All right. So if you see the screenshot, this is from the article which explained about that operation of that 3-parent baby. They tested the boy's mitochondria. They found that less than 1% carry the mutation, which can lead to issues. So this is too low, because to cause any problem, it is generally 18% of the mitochondria to be affected before the problem start. So that's what I said, right? That 18% you need it to be faulty, otherwise it's fine. But again, they will monitor the child's progress in his whole life. Now again, connected to this topic is designer babies or genetically modified babies, GMBs. So here you can see Parents can demand no baldness, they can demand high IQ, they can demand 20/20 vision, they can demand the child to be a sprinter and low risk of cancer and strokes, Alzheimer's, and etc. They can also demand perfect pitch, like to develop the kid into a singer, basically. Now, what are designer babies? Basically, these are those babies whose genes have been artificially selected and combined with IVF to ensure that the child develops in a certain way. They don't want the child to develop some issues, or they want child to have some features. So basically, they can design the child like you basically can design your car, like this color, this tires. When you modify, right? Many people modify their cars, many people modify their bikes, people modify their bikes when they want to go to Ladakh and so on, but that is obviously after you buy the bike. So this is before. All right, again, these examples that I'm giving you cannot be exactly compared, but they are very close examples. So here you can design the baby artificially. You can change the genetic structure of the child so that it has some particular genes or some particular features, right? So basically using biotechnology to have whatever type of baby you want. Simple, right? Basically the technology to design a baby is there, right? But this is basically used in animals as of now, not in the child. Three parent baby is basically, let's say, how it is different from designer babies. In three parent baby, you are actually solving the mitochondrial problem. So yeah, you are designing the baby to have better life. So that is why these topics are connected. There is not much difference. It's a designer babies is basically implication of mitochondrial replacement therapy. All right. Now DNA can be edited using the technique CRISPR-Cas9. We have already talked about this, right? All right. So this came into news around 2016 or 17 when a person from China, he claimed that he used CRISPR-Cas9 to produce world's first gene-edited babies. All right, it was in 2018, right? So what was the thing that was done? Basically the process. So this is a funny tagline: unnatural selection. Usually we study that there is a natural selection or natural order of things. Here it is unnatural selection. Basically it is artificial selection, right? So how to increase the chances of having a green-eyed blonde-haired daughter? So a woman's eggs are fertilized with sperm in a lab, creating several embryos. They will create a lot of embryos. One embryo leads to one child. So they will create a lot of embryos in the lab itself. Then a single cell is removed from each embryo and then they test it. Test it for what? They test it for biomarkers. Again, you don't need to understand this term biomarkers. Basically, biomarker is something that tells if you have that biological feature associated with the female gender, green eyes and blonde hair. So whatever embryo has these three markers, female gender, green eyes and blonde hair, they will keep that. All right, and they will take those embryos and then implant it into the mother's womb, and then most probably you will get a green eyed blonde hair female, right? So this is how you design babies. Again, you will remove or you will add something and then you wait for and then you check for biomarkers and then you implant it in the mother. Now one new technology related to IVF is IVG, in vitro gametogenesis. In vitro gametogenesis. Now, if we see the name, in vitro means basically outside the body, or you can say in a laboratory, or in a test tube, or in a petri dish. Petri dish. Gametogenesis is basically development of gametes, right? Or you can say from the germ cell. We studied about two cells, right? Somatic and germ cells, right? So gametogenesis is the process of formation of germ cells, all right, or basically the gametes. So IVF and IVG are different, very different. IVG is the future technology which is coming, and it is becoming famous because of the CRISPR technology. All right. So in this, basically, you don't need a germ cell from a person, that is egg or sperm. You don't need that. What you will do is you can take a cell from an adult person, any cell, and then reprogram it into a egg and sperm, and then produce a child. I will share a diagram, then you will be able to understand it better, but basic thing is this. All right. So IVF is an old technology. It started in 1970s. IVG is a new process that is still being researched. It has only been done on mice and not on humans as of yet. So this is the process. In IVG, you create a sperm and an egg from a lab from any adult cell, like from any somatic cell, right? You can use skin cells, blood cells, and you create induced pluripotent stem cells. I have talked about this. These are adult stem cells. So in IVG process, if it is applied to a human, right, you can simply give your skin, and then a lab would reprogram these skin cells into somatic into pluripotent stem cells. Now we have studied that stem cells can change into anything. So those stem cells will be basically transformed into egg and a sperm, and then the child will be produced. Now with this, you can create unlimited amount of embryos, and this will be very beneficial for same sex parents, like male female, same sex couples, especially for them it will be very beneficial. Also uh, it, uh, heterogeneous couples also would be able to utilize this. So IVG is a technology that is coming in the next, basically it's the next fertilization process. So this is the process which I just explained to you. It has already been tested on mice and not on humans. So this is the parent. You take cells from mouse, then you add genes to reprogram the cells, and then you create stem cells. Now these stem cells will be changed into egg, sperm and egg, and then they will be fertilized by IVF, and then you will create a offspring. Now this will not be a clone, but again this will have 100% of genetic material from one parent, right? So this is the IVG process. Now let's come to the next part of the lecture. Here I want to tell you the percentage of biotechnology firms in India by application, that is, in what field of biotechnology most of the companies are working. So the 51% companies are working in the area of health, right? The others are in bioinformatics, industrial processing, environment, food processing, agriculture, right? In India, biotechnology sector is quite booming, right? It has immense growth potential, and it will help in two things. It will help create jobs. It will make India manufacturing hub. All right. So we can also export and earn foreign currencies, right? Foreign exchange. So basically now I have related scientific thing to economy. So this is how you relate concepts between papers or even intra papers. Like in GS3, I connected economy and science and tech. You can also connect GS 1, 2, 3 and 4 with each other. This is inter GS connection. This is intra GS connection. All right. In health, biopharmaceutical sector accounts for the largest share, right? Basically, biopharmaceutical is the only sector. The health sector is the biopharmaceutical sector, and India is becoming slowly a leading destination for clinical trials and manufacturing activities. Also, in the case of COVID, we have manufactured 2 vaccines over here, and we have created a large capacity. Coming to the next topic, where domestic issues can be resolved with biotech, like biotechnology, where it can help India. So we have studied about the technology, we have taken about the science part. Now we are slowly moving into the mains part of the topic, where because this course is for pre and mains both. Once you do these notes, only you would be needing to update the current affairs, that's all. Obviously, we have covered the current affairs till now, but again, you need to understand the current affairs that will be coming in the upcoming months. So, domestic issues where biotechnology can help India. First of all, food. We need to feed our next billion. Also, this applies to the world also, right? Not just India. So, everywhere we need food security. And then second thing is food fortification. First, let us deal with food security. In food security, we need to make sure that people who are in need at least get 2 times of food, right? And how can we do that? By improving the crop productivity, by using GMOs, but using GMOs as safely as possible and with appropriate testing. Second thing is food fortification. We can increase the nutrient content of the food by using biotechnology. Like right now, food fortification is done. Like you might have seen some cereals like cornflakes, in which food is fortified. That cornflake is fortified by, let's say, vitamins. There are milk products also coming nowadays which are fortified with vitamins and minerals. So this is the thing which you need to understand. Then climate change. Climate change is coming. We can't ignore it. It's not going away. It will be here. So here, when it comes, we need to have crops that are resilient to climate change, or at least they can withstand. What will climate change bring? Unusual weather conditions, like sometime it will be very hot, sometime it will be very cold. Sometime the rainfall pattern will change. Sometimes cyclone will come. So the crop needs to be able to handle less water also and more water also. And we can also create crops which need less water. Like we can create a variety of sugarcane which needs less water. Then talking about environment. I've already talked about oil spills a lot. I've also given you a name of the bacteria. Go back and see that. Then there is radioactive waste. This is one of the most important things because a lot of nuclear waste is generated. If we can somehow create a bacteria which can digest the nuclear waste, then it will be wonderful for the world. Like I'm telling you, it will be wonderful for the energy sector. Because yes, I understand that nuclear energy creates a lot of waste, which is obviously harmful. It can change our DNA, obviously, but it is the cleanest source of energy that can be produced on Earth. Of course, excluding the renewable energies. So oil spills, radioactive waste, also other waste. If we can create a bacteria that can eat plastic, then of course, wonderful. Then health. I've talked about CRISPR, Cas9. They are making new therapies possible. Like in China, they did an experiment and delivered modified cells to a patient who was suffering from lung cancer. So they changed the cells in the lungs by using CRISPR-Cas9. Bioinformatics. In bioinformatics, you use the biotech and information technology. How? You use information technology to study the biotechnology, or basically, pardon me, you use information technology to understand biology. For example, the Human Genome Project, it would not have been possible without having powerful computers. So once you have mapped the genome of a human, you can create personalized medicine for them. Remember this, personalized medicine. So this is going to be a reality in the future. Then harnessing scarce natural resources. As we know, clean water is a scarce commodity. So as the population increases, we would need more cleaner water, more clean water. We would need desalination plants. So if we can produce some filters like biological filters, right? We can produce, let's say, a group of bacteria or group of fungi that can filter the water or filter the salt out of the water, then great, right? Which can, and we like I talked about remediation, like waste bioremediation. We can have some bacteria which can actually eat the, let's say, the dirt out of the water. So that would obviously benefit us. So there is huge level of potentiality that is there in this technology and how it can help a developing country like ours, which has such a huge population, right? Moving on to risks with biotechnology. There are ethical and political risks. In ethical risk, I've already talked about the consent. All right. So that is the first part. Then just a second. First, let me write that consent issue, right? So that you will be reminded when you see this slide. So first is the consent issue. Second is related with alteration of gene. That means whenever you change a gene in a human, that human will have some children and that those children will have their children. So basically you can potentially transfer harmful genetic material and you might not even know. The gene might change into something, but it might not be it might not be apparent to you. Let's say even if you're observing the child, let's say you have changed genes in some child and still you're observing them during their birth, but maybe the negative effect of the gene did not come into the, or basically did not come out during the life of the child, but it might get transferred or it might show up in the son or the daughter of the person. Then what? So basically you have, so getting my point, let me explain. If you have changed the gene of a person A, but the negative effect of the change does not show up in his own life, he lives the full life with no problems, but the problem get passed on to the child of this person, then how can you stop it, right? So this is the issue. So basically you will be changing permanently, you will be changing things permanently, right? Then intentional gene issue. Basically if you genetically enhance a person with some particular gene, right, that could, and people would want that, yes, I also want the same feature in my child. Let's say it's about athleticism. So if you count, then there are 30 to 50 features that everybody would want in their child, like not the full 30 or 50, but there might be list of 50 features and you can select 5 to 10 features and then you can have your child. So if you intentionally change the gene, right, so this will lead to loss of genetic diversity in humans, because humans like the fairer color, right? They might like blonde. Then what will happen? There would be less and less like dark tone humans, right? And which is not good for some people, because obviously that helps against that color comes from areas where there is a lot more sun. So people won't be able to have healthy skin in those areas like South Africa, even South India, right? It will reduce the diversity. See, what happens is nature changes things slowly, humans are changing things very fast. So the bodies will not have time to adapt, nature will not have time to adapt. So that is the thing, that is also the issue. So basically you will be destroying the diversity. Then come the political issue, basically data privacy. If you are giving your genome to a company to study your genes, what if it leaks? Right now we are facing data privacy issue in our normal things, like your phone number, your address. Your address is with so many companies now. Your phone number is with so many companies now. Virtually there is no privacy except the thoughts in your mind. Whatever you search get logged by Google. Logged in the sense LOGGED, like logged, right? Whatever you search on Google also gets sold to the advertisers. So privacy is reduced to a lot of extent. So but this genome thing will be devastating. If somebody has your DNA, they can, let's say there is a world political leader and his DNA gets into the criminal hands, so they can design a disease that will affect only that particular leader. So that is the gravity of the issue. Let's move on to the challenges faced in India. Challenges like funding. Only 0.67% of GDP is given to health research and overall health policies. Regulation. Under the Patent Act, Section 3D and compulsory licensing, due to which there are many companies which are not investing in India. Biotechnology Regulatory Authority Bill is also pending. There is third point is marketing, less public support and dependence on private funds. Basically, public is not supportive because they do not yet understand the full potential of biotechnology. So there is less support and dependence is on private fund, and you know about private basically companies, they focus on profit. So if they see profit in some technology, only then they are going to develop it. They are not going to develop a technology if it is not sorry, they are not going to develop a technology only if it is good for humans. They will develop technology only if it is beneficial for them. So dependence on private fund is also not good. Gestation period is quite long. That is lab to land. This is a keyword, lab to land. This also applies to agriculture. So gestation period is quite high in agriculture also, and this technology also. That means you have to do a lot of research before it can be created as a viable product which can be sold in the market. Return on investment is also quite low. Like if you invest a lot of your time, then the return on investment is less. Like you might not get a good job. So less quality jobs are there, hence less student put effort in this sector. Like less people are doing graduations and masters in this. Then there is lack of awareness, due to which many people oppose GMO also. Then there is brain drain. Other countries are offering much higher packages, much higher jobs, high quality jobs than India. So people are moving out and doing research there. What are the government steps that have been taken in this regard? The Department of Biotechnology in India, regulated by Ministry of Science, right? It has launched a National Biotechnology Development Strategy 2015 to 2020. Now, major initiatives under the National Biotechnology Development Strategy 2015-2020 is launch of 4 major missions in healthcare, food, nutrition, clean energy, and education. They have created a technology and translation network across India, basically in which universities from across India and foreign countries, they will combine and share knowledge. Then they have focused on building the human capital, basically you can say teaching them things, developing their capacity, increasing their skill. Indian government also set up a National Bioresource Development Board, right, which will determine the policy framework for efficient use of biotechnological research and development. That basically means they will, like I said, private firm will focus only on what it wants, but what might be good for the nation, they might overlook. So this board will look what is good for the nation and focus on that. They will guide policy with that. Other steps, National Biopharma Mission, the World Bank assisted Innovate in India. Now, aim of this mission is to develop new vaccines and therapies and new technologies in the biopharma sector. Then there is promotion of biotechnology in the northeast region of India scheme. See, whenever you're studying about UPSC, you don't need to study the details of the scheme. You just need to quote them. All right. Especially if the scheme comes, it might basically be asked that if it does this or not, or like it can ask if Innovate in India comes in National Biopharma Mission, like this. Now last point, the Indian government is also establishing many biotech parks and incubators. Some existing biotech parks are there in Uttar Pradesh, Hyderabad, Kerala, Himachal, and Bengaluru. So government is doing a lot. They have recognized the value of biotechnology and they are understanding that yes, we need to invest it so that we will reap the benefit in the future. So what is the way forward? We need to implement the Biotechnology Regulatory Authority Bill 2013. The industry needs to set up a roadmap for research and develop future plans, because with that the university will also try to link their curriculum. Industry, if it shows that yes, we need this, then university might also research. Government and industry collaboration, very important keyword, so that there is a balance between profitability and variety in research, because industry wants profitability, but for the nation, the variety of research would be beneficial. So government needs to fund variety in research. Industry can obviously focus on profitability, but we need to combine these two forces and then create better research. We need to have better human resources by teaching them better things. We need to provide them better practical training, on-site training. Then of course we can have funding, but here Don't just write funding. You can use this point anywhere, wherever you need to write about funding. Have a competition based funding, right? Of course, you can't do this for schemes, but such things for science and technology, you can write this, that funding should be competition based, so that you can have the best ideas and then put money in that. So chances of success are higher. So remember, for social sector schemes, don't write funding based on competition, because welfare, India is a welfare nation. But for science and technology, and where we have limited money, you can write this funding based on competition, and that would be enough. The evaluator will understand. So one interesting thing I would like to discuss is the future of biotech. Now it is quite bright. All right, it is quite bright because of the recombinant DNA technology, because of which you can basically engineer new microbes or bacteria. You can create new plants, you can create new animals, so that they have new capabilities which have not been seen yet. Then we can create genetically modified organisms, right? And this is basically from recombinant DNA technology only. And this has been helpful in what? Increasing crop yield. It has reduced post harvest losses. It has created stress resilient crops. So lot of benefits from recombinant DNA technology, right? So basically, to let's say, to reap the full benefits of biotechnology sector, the biotechnology industry should continue to educate people. Regulators need to form regulations that are basically updated with time. It's not like you make regulation now, it will update it after 5 years. No. All right. So basically, the challenge that is faced by humanity is of change. We need to quickly adapt. As Darwin said, people, the species which adapt survive. So we need to adapt, because there are a lot of challenges coming of feeding the next billion, climate change, right? So in future, I'm pretty sure the people will ask question that why aren't we using biotechnology to solve this problem? Why aren't we using biotechnology to solve climate change problem? Right? Right now, people are asking what is biotech, but I'm pretty sure within 20 years, this statement will change to why hasn't biotechnology solved this problem yet? So that is how I feel the future is bright of biotech. Now, the last point for this lecture, protocols regarding the biotechnology. Now, this is already, I hope this has been discussed in your environment sessions. If not, then it will be. But these are the part of your environment lecture. So, Cartagena protocol on biosafety. Now, see why it is there. Biotechnology can lead to negative results also. It has a capacity to threaten biodiversity. Like if you introduce a alien gene in a new land, let's say you introduce a tomato that was not native to India, right? Or you change something which is not native to India and you bring it to India. So it might affect the biodiversity of India, right? So invasive species, what I'm talking about. So to protect from such things, we have this Convention on Biological Diversity. So it has biosafety protocols to protect the biodiversity. It basically regulates genetically modified organisms. Now the Nagoya Protocol on Access and Benefit Sharing, ABS, very important thing. So here, equitable access and benefit sharing is focused. Whenever benefit arises from accessing genetic resources, it needs to be shared with the local community also, right? All right. So that is all for this lecture. I hope you have understood things. If there are any doubts, please let us know and do revise, because as you know, tests are coming for you and this is important section. Biotechnology is quite important. There are a lot of new terms, there are a lot of new technologies. All right. So if there is anything, please let us know. Till then, take care, do revise, and see you in the next lecture. Bye bye.